This video is all about fermentation. I've mentioned before that pyruvate is a major branch point in cellular metabolism. Cells can take pyruvate, import it into the mitochondrion, and feed it into the TCA cycle. Pyruvate also serves as the starting material for the synthesis of some amino acids, such as alanine. And pyruvate is also the beginning material for what's called alcohol fermentation and lactate fermentation, and those two will be the focus of this video. Well, let's look at them one by one, beginning with lactate fermentation. Well, the starting material is pyruvate, and the end product is lactate. They're both 3-carbon acids. The enzyme that catalyzes this reaction is called lactate dehydrogenase, and that name should suggest that it's a redox reaction, and it is. And in this case, what's happening is pyruvate is being reduced to form lactate, and it's coupled to the oxidation of NADH to form NAD+. We can often recognize redox reactions by the appearance and disappearance of a hydrogen, and that's true in this reaction. Now, you might be a little surprised that cells want to carry out this pathway because I've told you before that NADH represents a source of energy in the cell, and here we're consuming it. So why would a cell ever resort to lactate fermentation? To answer this question, we need to think about the big picture. So remember that when we carry out glycolysis, glucose is being broken down in order to form pyruvate, and in order to carry out this reaction, we need to have also ADP and NAD+. The products include ATP and NADH. Well, cells are always using ATP. They use it in a lot of different metabolic pathways, so ATP is constantly being broken down and forming ADP. The other product, NADH, is used in some cellular pathways, but most of it's oxidized in the electron transport chain, and that process requires oxygen. So if there's not a lot of oxygen around and the cell is unable to do this, what will happen is NADH will accumulate in the cell and it will deplete NAD+, and that would bring the whole glycolysis reaction to a standstill. In order to regenerate the starting material, what cells do is fermentation. They'll take the pyruvate, convert it into lactate, and this is going to allow them to regenerate the NAD+ needed to keep this process running. A cell running glycolysis and fermentation will be able to produce small amounts of ATP and thus stay alive. Let's look at alcohol fermentation now. Alcohol fermentation is a 2-step process. In the first step, catalyzed by the enzyme pyruvate decarboxylase, a carbon dioxide is removed from pyruvate to form acetaldehyde. The second step is catalyzed by alcohol dehydrogenase. This is a redox reaction, and once again, NADH is being oxidized to form NAD+. So a cell that's running glycolysis and alcohol fermentation will also be able to regenerate the NAD+ needed to keep the cell going. Let's look at the products of the reaction. For alcohol fermentation, the first product is carbon dioxide. I've told you before that carbon dioxide is small and very membrane permeable. It will diffuse away from the cell. The second reaction yields NAD+. Well, we know what's going to happen with that. That will feed back into glycolysis, get converted into NADH, and this cycle can keep on running. The final product is ethanol. Ethanol is also small and pretty nonpolar, so it's able to diffuse through cell membranes and exit the cell. Under normal circumstances, human bodies don't carry out alcohol fermentation, but we do use alcohol fermentation in cooking. For example, in bread making, the yeast in the bread carry out alcohol fermentation, and they create carbon dioxide, which allows the bread to rise and get puffy, and ethanol. During the baking process, the ethanol will be baked off. Here's a different example of humans using alcohol fermentation. This is beer making, in which yeast are added to a solution containing carbohydrates, breaking them down, and as they do this, they produce CO2 and ethanol. What happens to the waste products in lactate fermentation? Well, once again, this reaction will produce NAD+, and we know what will happen with it. A cell running glycolysis and lactate fermentation will cycle the NAD+ and NADH between those two reactions. The other product, lactate, is charged and it will not pass through the membrane on its own. It requires a membrane transporter called MCT4 in order to exit the cell. In mammals, the lactate is recycled, so that once lactate is released from the tissues, it can be reabsorbed in the bloodstream and then transported to the liver. Liver cells can run lactate fermentation backwards, taking the lactate and converting it into pyruvate, and that gives animals a chance to recoup some of the energy that they might have lost by bypassing the TCA cycle. Diving mammals such as seals provide a dramatic example of how lactate can be recycled. This graph presents us some classic data on what happens to the levels of oxygen, carbon dioxide, and lactate in the blood of a gray seal before, during, and after a dive. I know it's a little bit complicated, but let me try to walk you through it. What we see before the dive is that there are very low levels of lactic acid. That's the yellow line labeled LA. When mammals breathe air, the lungs are very efficient at oxygenating the blood, and so if we look at the relative percent of oxygen in the blood, it's equivalent to what it would be in the atmosphere, about 20%. As animals consume oxygen, they're producing CO2, so the CO2 levels in the blood seem high, but they'll be exhaled at the lung and removed from the body. Now let's see what happens during the dive. So here's the dive, 18 minutes long, and what I want you to notice is that as soon as the animal is underwater and no longer has access to oxygen, we start to see a drop in the level of blood oxygen levels. There's a corresponding increase in the levels of CO2. That should make sense. The animals are initially continuing aerobic metabolism, using up the oxygen, producing CO2. After a while, we start to see a modest rise in the amount of lactic acid in the blood. This is showing us that the animal is switching and relying more and more on anaerobic metabolism. When the dive is done, and the animal is able to come up and get air again, we see something amazing. There's this huge spike in the amount of lactic acid in the bloodstream. What's happened here is that all during that dive, the seal muscles were making and accumulating lactic acid, and then once the animals return to the surface, they dump that lactate into the bloodstream. It can be carried to the liver, and then the reason that that yellow line slowly falls is because the liver takes that lactate and converts it into pyruvate. This way of dealing with lactate's not unique to seals. If you go out for a run and you're exercising, you'll produce lactic acid in your muscles, and after the run, that will also be sent to your liver and converted back into pyruvate. So the next time you're exercising, or maybe having a piece of bread, think about the role of fermentation in making that possible.